today. My name is Ed Creedon. I'm a senior project manager with AST Environmental. The AST is a member of the RPI group, and I can share a little bit more with you about the RPI group in a few moments. But first, I'd like to talk about today's presentation. It's going to be given by Dr. Ed Winner. Ed's the Vice President of Remediation Products, Inc. And he will be speaking about the RPI activated carbon-based technology known as BOS 200 Plus. Now 200 Plus is an enhancement of the BOS 200 product, which has been used on petroleum release sites for nearly 20 years. And although BOS 200 Plus was developed more recently for use at sites with high concentrations of contaminants, including LNAPL, it's really now being used at most petroleum release sites for a variety of reasons. One of which is that quite often we do discover during project implementation that there's higher concentrations than were expected, even LNAPL at sites that seem to be a little bit less impacted. And it also has the ability to further accelerate the bioremediation process and get your sites to closure even faster. So Ed's got a lot of really interesting information to share with you about this technology, how it works, and he's got some performance monitoring data to share as well from sites where 200 Plus has been applied. Before I hand it over to Ed, I want to take a quick moment and tell you more about the RPI group. So the RPI group, of course, includes RPI, the developer of the BOS 200, 200 Plus, BOS 100, and CAT 100 products, along with a network of authorized installers, and that includes AST. AST is also responsible for RPI project assessments and design, the field quality control, and we are also the worldwide distributor for the RPI products. RPI is headquartered in Golden, Colorado. AST is headquartered near Lexington, Kentucky, in Midway, Kentucky. I'm in Kansas City, and other AST locations include Denver, Philadelphia, Virginia, and Ohio. And our international RPI group members are located in Northern Europe, Canada, and Australia. So when we talk about cleaning up the world one site at a time, we truly mean it. Now, if you have any questions about the field methods for product installation, that's not something Ed's going to go into much detail on today, or how we apply the various RPI technologies to different contaminant sources. You can always visit the AST or the RPI websites and reach out to me or Dr. Winner at any time. And the websites are for RPI, trapandtreat.com, trapandtreat.com, and AST's website is astenv.com. And of course, you can find our contact information there. Okay, so let's get to the good stuff here. Remember, you can post questions throughout the presentation using the Q&A tab in your webinar control panel. I'm assuming you are all very familiar with that process now. And we will have time at the end to answer your questions. So, and also keep an eye out for a recording that we'll distribute in the coming days of today's webinar. And also, by the way, the RPI and AST websites have great libraries of recordings of previous webinars about all these technologies and applications. So be sure to check those out if you can. So I'll be back after Ed wraps up his talk to lob your questions his way. Okay, Ed, please do take it away.
Okay, I'd first like to thank everybody for being with us this afternoon. What I want to talk about is what is different with the BOS 200 product. And in order to do that, we'll start with some contrast. I should say I should have built a more interesting title, but I think that this is a very practical title. So let's go and look. Okay, both BOS 200 and 200 Plus are based on activated carbon, powdered activated carbon. They're supplemented with electron acceptors, nitrate and sulfate, and they include microbes, which are bacteria, yeast, and fungi. So that part remains the same. BOS 200 Plus adds supplemental nitrogen for anabolic processes. So understand that the nitrogen that's supplemented as an electron acceptor is not necessarily nitrogen for anabolic processes, so you need more. It's not that they aren't interchangeable, but you need additional. Supplemental carbon for energy production and anabolic processes. And BOS 200 adds additional microbes to augment 4 characteristics. We want to break down supplemental and endogenous carbons to base sugars or alcohols or whatever it happens to be, but the provided or supplemental carbon goes to base sugars, additional sources of vitamins, additional measures to ensure no hydrogen sulfide buildup, and we wanted to be sure that it was robust in the presence of LNAPL, and that's why we've added this supplemental carbon, vitamins, supplemental nitrogen, etc., and the microbes to break down the supplemental carbon. The core element of both products is the activated carbon, and this is important. The structure of the activated carbon gives significant absorptive capability so that you can pull the hydrocarbon into the activated carbon. And the absorptive force is the sum of all the individual interactions between the carbon atoms and the absorbate molecule. So if you look at this, looks almost like a hamburger picture in blue. These are various macropores that you're seeing on the surface of the carbon, and micropores, which are deeper into the carbon primarily, would be that absorptive surface where you retain the hydrocarbons, and that helps you deal with the actual concentration that is in the aqueous phase. Some general points about carbon grind. So we're using powdered activated carbon. There's also a granular activated carbon, which is much larger. The ASTM standard is 180 to 101 micrometers. The point of the showing you the different grinds is so that you can just be aware of them. The colloidal carbon, which you hear quite a bit of advertising about, is down to 1 to 2 microns. There's no set ASTM standard for it. But nevertheless, we use the powdered carbon. We think it gives us the best balance between distribution and another aspect, which is what you're seeing here in this picture. Carbon does not just absorb. Carbon can become, if you select the right carbon, a platform for microbial growth. So if you look at the picture on the left, this is clay material under an electron micrograph, and the red circle is circling a single bacteria. And I'll take a chance and use the little spotlight here and say, see, that's circling a bacteria. Here's another bacteria. Here's another one up here. This is on clay material. Look at the difference by the same authors using the same methodology on activated carbon. You can see the bacteria here and then all of these bacteria. The point is that the activated carbon is a much better surface for microbial growth than is, let me turn that spotlight back off. There we go, is a much better substrate for microbial growth. If you have a better substrate, then you can sustain higher microbial counts. Higher microbial counts, all things being equal, is higher degradation rate. We provide electron acceptors. This shouldn't be surprising to you. It's well known in the industry. Sulfate and nitrate are typical. If you provide enough sulfate, you can restrain some of the methogenesis that can occur. I happen to be pointing out in this case disulfibrio, which can use nitrate, but actually prefers the less energetic sulfate. It's kind of interesting side note to kind of keep in mind, because it kind of, I think it helps you put some of this in context. The carbon to nitrate balance So we're adding additional carbon. There are lots of reasons we can do that, but at least one of the reasons is that your carbon to nitrate needs to be in a range, a ratio. So your label carbon needs to be 15 to 45 times that of your dissolved nitrogen. So if you just decide to go out and I'm just going to add as much nitrogen in the ground as I can, what will happen is the microbes will shut down. There's an entire system, feedback loop controlling how nitrogen is metabolized relative to the amount of carbon that can be taken up. So you can't really just take the agricultural approach and say more fertilizer is better. Now, how can you play a game with that? Well, if your labile carbon is low, and hydrocarbons are very typical, they're not labile, you've got a problem. But what you can do is you can add labile carbon, such as starch, and that allows you to increase the amount of nitrogen. Now, what happens with that nitrogen, a lot of it gets tied up in the microbes that grow. So as you get a larger and heavier growth of microbes, those microbes take that nitrogen and they turn it into more labile forms, not just of carbon, but of nitrogen, such as amino acids, proteins, etc., which when they die are available to the other microbes. So carbon availability itself exerts a strong control on bacterial nitrogen dynamics. A high carbon to nitrogen ratio, when the carbon is largely refractory, results in low bacterial abundance. And this is the case for most hydrocarbon sites. Simply adding more nitrogen won't work. Organic nitrogen is used more than inorganic nitrogen when the label carbon concentrations are low. So you have to be careful at that point exactly what nitrogen you're putting in. However, if the labile carbon is relatively high, then the microbes don't discriminate against the forms of nitrogen. That gives them a broader ability to use the nitrogen available in the environment. Starch itself is a very rich carbohydrate that can be broken down into base sugars. These can be easily used by fermenting bacteria, and the supplemental bacteria that we add have a purpose of actually breaking down that starch and releasing hydrogen and CO2, while the major part of the carbohydrate is actually converted to volatile fatty acids. Now, although long-chain fatty acids and proteins themselves can produce hydrogen, and a typical long-chain fatty acid itself, or something very similar to it, are some of the alkenes, only the fermentation of carbohydrates to hydrogen is thermodynamically favorable. So you're also driving the reaction from a thermodynamics standpoint. The additional microbes that we've talked about are bacteria, yeast, and fungi. Their purpose is to break down, as I've already mentioned, that supplemental carbon, to add a source of vitamins and amino acids, to restrain hydrogen sulfide buildup, and potentially, one would hope, and we have seen it to be so, that you would diversify the xenobiotic pathways, the degradation pathways. So let's examine the microbial impact of BOS 200 plus. Now, keep in mind, I'm going to focus primarily on anaerobic degradation. So let's say we had a PAH. A lot of this we'll be talking about will be alkenes, but nevertheless, you need to somehow activate it by hydroxylation, carboxylation, methylation, etc. And then you can either go an aerobic pathway or the anaerobic pathway. I will just say, just to make it clear, if you have a site where you could actually sustain an aerobic environment, well, you probably wouldn't have that site because it would take care of itself. But if you could, that's a good way to go. Most sites, because of the presence of quite a bit of contamination, tend to be anaerobic. The stock pathway for anaerobic degradation is called beta oxidation. There are other pathways, but this is the way this particular slide was set up, and beta oxidation is the main pathway. Anaerobic metabolic scheme. You start with some particular organic matter. It could be the carbon we've added. It could be the breakdown of a hydrocarbon, a protein, a lipid. You hydrolyze it ultimately to sugars, alcohols, fatty acids, all of which can be fermented. Those fermented products will eventually head on to methagenesis. So if you have a low redox potential below 200 microvolts, negative 200 microvolts, is necessary for efficient methagenesis. So if you keep that high by adding oxygen, okay, you wouldn't go this direction metabolically anyway, but you could use sulfate, nitrate, etc. So this is the standard kind of anaerobic metabolic scheme of hydrolysis, acidogenesis, acetogenesis, and methagenesis. We won't spend much time on it, but I thought I would lay it out for you. Now, this is data from a heavy hydrocarbon site, 1 year and 16 months post BOS 200 injection. I used this site first because I just like it as much as anything. The idea that it is just a very heavy hydrocarbon. It's not your typical gasoline site. And if you look on the left-hand side, you see the impacted control. So this is an area that has not been treated. This is what it looks like. You're almost entirely Smithella. You look 1 year out and look at how broad the microbes have become as far as who's present. 16 months out, you've yet increased that again. These are not all the microbes that are present, but I stopped right at about, 0.5 or so to make the picture look even. So let's focus on the 16 months post BOS 200. And this is a very complicated slide, and there's more to it than I really intend to cover. But if we look on the left-hand side and look at Smithella, Smithella is big on degrading alkenes, and it'll degrade those alkenes down to CO2, hydrogen, formate, and acetate. So it's also a methagenic bacteria in that sense. And you see that when you come to the site, the impacted control is driven by Smithella. And then you have two other microbes, which I'll emphasize, Candis sulfocarvium and Methanoregula formica. I mispronounced that, but nevertheless, there it is. Those two are dealing with the products from Smithella. So you don't really have, in this case, a very strong or deep set of microbes. You're pretty much breaking down into hydrogen CO2 and doing hydrogen CO2 fixation. Now, if you look out at 12 and 16 months, you start seeing clear fermenters, and I marked those by beer bottles. I may have missed a few, but nevertheless, it gets the idea across that you're now doing fermentation. You're now doing, and you were before, I just forgot to put the table with the little characters there. The table with the characters are doing degradation. And then I marked in some cases whether you're doing sulfate reduction or nitrate reduction or denitrifying bacteria. The gist being that now you have this rich population that can do more than just direct methagenesis. Ooh, this really becomes complicated. Okay, now what's going on here, and this was stolen from down here from Embry as far as the Let me get myself a spotlight again. That's an awful big one, but nevertheless, we'll use it. It was stolen right here from Embry, so you can look at this paper. It's a really good paper, actually. And we're going to use hexocaine here as a model, but and this is specific for hexocaine, but it could be any alkane that would be broken down by Smith-Hella. And ultimately, you're going to produce acetate, formate, hydrogen, and electrons. You have two routes that were emphasized, at least in that particular journal article, because they were talking about the methogenous routes. The hydrogenotropic route, where you're taking your CO2 and hydrogen, and the autotrophs are converting that CO2 to biomass and using the hydrogen as an electron donor. And you will see that the site that we're talking about, this heavy oil site, went almost fully in this direction that I'm showing you with the pointer. You have some formate and hydrogen going to you use sulfate and as your electron acceptor. You have some other microbes that are using nitrate. It's not Disulfosporcinus, which is of course using sulfate. But this gives you a really interesting kind of picture to keep in mind of what is actually occurring, that you're breaking these things down, that the formate and hydrogen can be released into the extracellular space. It can be picked up by other microbes and processed. You will see later on when I go to a second site that the second site went primarily this direction towards the acetotropic route with acetate and electrons. The methanotrex solingeni is not present in the heavy hydrocarbon site, but it is present in the other site I will show you, and you'll find it that has pertinence to you. Okay, so what I'm trying to emphasize is you have multiple routes of metabolism. In this heavy oil site, you are doing sulfate reduction, nitrate reduction, denitrification, and methagenesis, all in a rich population, a population that was far richer than what you started with in the impacted area. Now, if we look at some typical measures of ecological abundance, and we'll start here at the top with the CHO1 right here, the CHO1 abundance index. Now, what this index does is it weights those microbes that are present in very, very low numbers, ones and twos like that. And you would think, well, that's really odd. Why would I care? I'll get to that in a moment. But you see that post BOS 200 plus, you have a much higher CHO index than you did either in the control or the impacted control. The Simpson Diversity Index measures evenness, and you could see why this would be important. So if I had 90% of one microbe, then my evenness would be very low, right? And what you would really like, or you think you'd like, that in a environment is that microbes have fairly even distributions and fairly even numbers. I think that the Simpson is not quite as useful, to be honest with you, because you can easily produce a lot more byproducts from a microbe like Smith-Hella, such that you can have smaller numbers of the microbes downstream, such as your acetogenic methanogens. So it's not quite as good a measure, but it is a standard measure. The next measure is the Shannon-Weaver. This is a good measure because you start balancing the richness, means the number of microbes in total, the various species, and your evenness. And in each of these cases, you see that post BOS 200 plus injection, you have an increase. Now, the way I would think about this is the idea of richness, just plain having more species itself, all things being equal, you're going to get higher degradation. The other aspect is if you actually increase the number of unique individuals, you think about like a basketball team or something, and they'll say, hey, they've got a really deep bench. And that's kind of what you're looking at. When you have a high CHO, you kind of think of having a deep bench. And that's important because as degradation occurs over time, and as you go through seasons, sites change. Some aspects of your hydrocarbon, some molecular or chemicals within that hydrocarbon are much easier to digest than others, and they will be digested first. They like to eat their dessert before their dinner. And so those will go first, and you will see the ratio of the various contaminants within the hydrocarbon mix change over time. So you need a deep bench. Now I'm going to go to a different site, and this site happens to be called Bethel Springs. Now, what this graph is showing you, or what I want you to see from this graph, is which microbes jumped up as being unique during the process. Okay, so if you look at the very top in green, you'll see the pre-injected area to be treated. Okay, so these are the areas we're going to treat, but this is what they look like pre-injection. And you would see Extensomonas perlucida being very dominant and Geobacter metalloreductans being very dominant. If we went all the way to the bottom and looked at the blue stripe right here that I'm underlying, this is impacted background, which we didn't treat. And we see Desulfocurvis vaccinus being dominant. What that's telling you is in the background right now, it's doing sulfate reduction. At least the dominant bacteria that stood out as being unique to the other bacteria that were present in the background. Now, microbes in the unimpacted background were very broad, and you can see them. Gallinella would be one here at the top, etc. And then immediate post BOS 200 plus injection, you see Sulfospirillum caervole jump up as being important. When we get into the second quarter, you see the Desulfobulbus as being of significant importance, Desulfomaculum profundi, etc. Okay, so all of that is all very interesting, but what is the point? The point is that the process of being impacted or not being impacted, being injected on time, each of which are modifying and changing the microbial population in a way that can be picked out and pointed to statistically. So when you look at the second quarter here, you see the desulfos jump up. Anything that starts with desulfo is basically a sulfate reduction. And we're putting in sulfate, so it shouldn't be surprising. So are you having an impact on the population based on the effort that you're taking? Yes, you are. Okay, now this is also a very complicated looking picture, but it's simple enough if you give it a moment. I tried again to mark the degraders with some people eating at a table. Some of the others here may also be degraders. For example, Extensomonas perlucida can do degradation if a different microbe does activation. So it has many of the genes for downstream functions. It just doesn't have any for the upstream functions, which tend to be more important. The other thing I want you to notice in this, besides the rich number of degraders, is that each of the monitoring wells moving left to right look somewhat different. And in terms of the microbes that are expressed and the numbers and percentages they are expressed in. So if you look at the blue arrows, I'm going to start with the first blue arrow on your left, which is, in this case, monitoring well 5. And in monitoring well 5, you see Smithella is approximately, we'll call it 27%, and Disulfobulbus toluene SR at approximately 27%. What this is telling you is, in this particular case, you're doing sulfate reduction, and you'll actually see, if you look down, you see very few microbes that are involved in methogenesis at this point. Monitoring well 5 is very close to, on the far right, RW3. And it's not surprising that they look very similar. That Smithella is in high percentage, 37, 38%, and Disulfobulbus toluene sulfate reductase, or sulfate reducing, is like 28 or 27%. Now, if you look in the middle between the two, you got monitoring well 7. Now, it's also very rich in Smithella, but Disulfobulbus is much lower, where Methanotrex solingini, which is at almost 18%, it's the other end of the arrow, is quite high. And this is an acetotropic pathway. Now, remember when I showed you, and I'll go back up just to do it, I showed you this overly complicated picture here. And on this picture, I showed you that the first site, the heavy oil site, tended to take this route in what methogenesis it was doing. But now I'm showing you a gasoline site, which is also, it's an LNAPL site, is tending towards this direction. Now that is actually indicative. If you could look at the microbes on your sites, the sites that are contaminated with the heavier oils, will almost always, like, oils, waxes, that kind of stuff, will favor this route across to the top, the hydrogenotropic route, versus the acetotropic route. Okay, so let me close this pointer out and move back to the slide we were on. Here we go. So I thought that was quite interesting. It's also interesting that you see within that same monitoring well 7 that the Pseudomonas jumps up relative to what you're seeing in Smithella, which you're seeing in monitoring well 5 with Smithella and Disulfobulbus. And in all likelihood, that's also indicative of what you would see if you looked into the particular chemicals that are being expressed, are being monitored for. Boy, I'm not doing that well. Let's try it again. If you looked at the analytical data in the monitoring well, you would see that monitoring well 7 looks very different from 5 and 3. And that is expressed in the microbial population. There you go. Let me wake up. Okay, next slide. So in this particular case, and on the heavy oil site, both cases, we increased the number of cells. So if you look at monitoring well 1, which is impacted and not treated, versus monitoring well 12, which is impacted background, compared to the treated areas, monitoring well 5, 7, 14, and RW3. You'll see that the cell numbers increase and are more consistent than that which you see in either the impacted but not treated or the unimpacted background. Now remember that this is on a log scale, so little bit of differences are big differences, right? So what have we covered? That BOS 200 and 200 plus, we're still using the powdered activated carbon. We still believe that that is the best route because anything smaller just does not provide space for the microbes to dwell on the surface and doesn't provide microbial support. Larger is harder to install. We give supplemental electron acceptors of nitrate and sulfate. We're now able to add more nitrate, and we explained why earlier, and we still have microbes, bacteria, yeast, and fungi. BOS 200 plus adds supplemental nitrogen for anabolic processes, supplemental carbon, which allows us to yet add more nitrogen beyond the anabolic processes, and additional microbes to break down the supplemental carbon, to add additional sources of vitamins to ensure that we have low hydrogen sulfide buildup and ultimately be robust in the presence of LNAPL. So we want to build microbial populations that logically conform to expectations. And that was a lot of what I was trying to show you in the microbial, showing you how the microbes interacted with one another, okay? That this is logical, that this is consistent, and that they have metabolic functions that complement one another. We also wanted to ensure that we have a more diverse microbial communities, a deep bench, and I showed you that that's true. And we want to sustain higher numbers of cells over time, and I've shown you that that was true. Now, this is an introductory presentation to a presentation that Bill Brab will give, where he actually goes into the site geology, the injection of the site, and what the monitoring well data is showing. But in general, the monitoring well data is good. It's consistent with that which I've shown you, that we are building these communities, we have a deep bench, and we are sustaining the numbers. Okay, Ed?
Okay, thank you, Ed. Excellent job, as always. I know I always enjoy learning about these technologies and the changes we see in the microbial populations over time. There's a lot there. We do have some questions here, and the first one here asks that please describe the importance of archaea, if I'm pronouncing that correctly, in the biodegradation process in your two studies.
Yeah, the archaea are primarily methanogens. What you'll see is if you look in the, I don't know that I marked them, but you don't see a lot of archaea in the, look at some of the larger lists, you don't see a lot of archaea in here, and I didn't mark them. So sometimes maybe somebody's better than I am and they can just pick them out by name, but I can't. I usually have to mark them first. But over time, I mean, if you're just allowed the site to just go straight into, into methogenesis without electron acceptors, you'd see the archaea number being much, much higher.
Okay. Hey, Ed, I noticed on your last slide where you had your contact information, there was S missing in that. Ed at Remediation Products, plural, products.com. So point that out to everybody there in case they're getting a bounce back from you. Okay, we've got some other questions here. Once the cleanup is complete, do the background bacteria repopulate the area?
You know, I don't know that we have followed a site long enough for me to say definitively the background bacteria repopulate the area. In a sense that, yes, it's all those guys again and no one else, right? But from the time that we have followed, I do see that as sites, lose the hydrocarbons, as the hydrocarbon concentration goes down, that the population starts to look more and more like the controls. So I would suspect, though I can't tell you on what time frame, they would eventually return to either the exact same population or populations that were very, very similar.
Sure. Okay. Here's an interesting question. I've had this tossed my way before. Why is activated carbon, as in BOS 200 plus, a better medium for microbial growth than subsurface soils?
Yeah, and I think we can go back to the picture. I'm going to go page up here, just kind of confirming that observation. And you can, for the clay material versus the activated carbon, and you can go through the literature and see, you can see many, many pictures like this. In fact, I can't think of a single material where you see the numbers or the heavy growth that is anywhere comparable to activated carbon. Part of it has to do with that activated carbon itself is adsorptive. So you are drawing potential food sources into the carbon. That might be your hydrocarbons, but it may be, in fact, it is your hydrocarbons, right? You see your hydrocarbon concentrations go down, they're going into the activated carbon. And it's other things that are within the subsurface that might be edible. That's one aspect. The other aspect is that the activated carbon itself, the macropores, are sufficient for microbes to kind of duck down into to provide them a space to initiate growth and find protection. We don't really think a lot about microbes needing protection, but they do. There are a number of other larger microbes that graze on them, that eat them. There are also smaller things such as phages, which attack them and essentially, in human terms, make them sick, right? Kill them. So there's need for protection. And there's also a need to have to gather in large numbers. So if they can gather in large numbers on a surface that is conducive to binding, then they can share metabolic products. The classic understanding of that is that they build biofilms, and the biofilms themselves are made as much as anything out of sugars. And so in that biofilm, they share products with one another, they share protection, and they would be able to change actually concentration ratios across the surface of the carbon such that they can draw nutrient from the carbon. It's also true that activated carbon holds some degree of water. So if you think about in the smear zone where the water is bouncing up and down, the microbes that are attached to the carbon are going to sustain that changing water scenario much better than microbes that are on some other surface. So those are three reasons. There are others, but I think those are three good solid reasons.
Sure. Yeah, and I also would want to point out to everyone that Dr. Winter gave a presentation that has some excellent photographs of these biofilms and the biological growth just not too long ago, and you can find that webinar on the RPI website. It really addresses this question very directly. So I've got a couple questions here that are really related. Kind of one is asking about the importance of microbial diversity. Why is it important? And then it really ties into another question where One of our guests here asks, does the new consortium break down more contaminant or better reduce the contaminant concentrations? So I think, talking
Yeah.
about the original.
No, I got it. The reason I paused is break down or reduce. I suppose it's possible that those two things could be different, but we'll just assume that the questioner meant those to be the same. Yes, the new consortia, the larger, deeper benched consortia, break down more hydrocarbons than you would see. Well, certainly than you would see in the impacted area, right? So that's the gist. I guess a good way to think about this is in every ecosystem, you have certain key animals, okay? Some people call them keystone species or something like that. So, if you're wolves, you need a lot of rabbits to eat. And if you don't have enough rabbits, then the wolves die. If you have too many wolves, then you don't have enough rabbits, et cetera, and you try to maintain that balance. Well, you could imagine an ecosystem that was nothing but wolves and rabbits. It would be constant boom and bust cycles, right? When all the wolves died, the rabbits would bust. When all the rabbits bust, then the few wolves would go up. So that's what you're trying to avoid, because the subsurface itself is an ecosystem. It's an ecosystem for microbes that spend most of their time in starvation mode. So you'll go through, late fall through most of the winter months. The groundwater is cooler. The sources of food have dropped off. And sometimes, depending on how the water table moves, you have lost the transport system that's bringing you these things. Then spring comes along, and what happens in spring? Oh, you get spring rains, you get plants are starting to bloom, the roots are releasing sugars and various other exudates, and what happens? The microbes are going, oh my gosh, wonderful food, right? And they bloom. And that lasts through part of the summer until you start to get into the dry season, and then they start to taper off again. Then fall hits, the leaves fall, the leaves degrade, all of that goes in the ground, and the microbes boom again, and then they drop off. So this is the lifestyle they're used to, this kind of starvation lifestyle. And what we're trying to do is to take that and increase the numbers and hold the numbers at high levels. In order to do that, you can't depend on just a few different microbes. You need as deep a bench as you can to deal with the changing environment. And the changing environment in this case is you are degrading hydrocarbons that are easy to eat, and then you're going to move on to hydrocarbons that are harder to eat, as an example of one aspect of the changing environment. So you need that deep bench to sustain that degradation. Now, it's also true that there's some toxicity that's going on, because some of the microbes will eat part of a thing and spit out what they don't like, so to speak. I'm being very simplistic about it. I apologize, but it's easy to keep the picture in mind. So you have to deal with that too. You need the microbe that says, oh no, one man's trash is my treasure, who wants what the other one is spitting out. And that again is found in a deep and broad bench of microbes being present. Boy, that was too much, wasn't it, Ed?
No, well, I don't know. I stuck with you through most of it. So,
Okay.
okay. So we've got a couple more follow-up questions here from Ben. One is, are polar compounds produced?
I'm kind of stumped because I'm not sure.
Yeah, I'm not sure.
I mean, fatty acids are polar compounds, right? So yeah, they're produced, but I don't think that's what the questioner meant or intended, because that doesn't, well, I guess it's yes.
Okay. Well, and Ben, I'll just say, feel free to reach out directly to Ed. He will gladly speak with you about this
Oh
later
yeah,
too.
I'd be glad to. I probably just, sometimes I just don't follow where another person's coming from, and you'll tell me and I'll go, oh, and bounce on my head, right?
Sure.
Okay.
Okay. And then finally, he asked, is the production of more methane considered a contribution to the global warming problem?
That's a great question, because you do have to think about it. But as you move up higher into the surface of the soil, there are microbes that feast upon methane, so that if you were to actually look at methane close to the surface, you can see it increase. That is true, but it wouldn't increase to the degree of the amount of hydrocarbon you're actually degrading because of the methyl that are in the upper surfaces of the soil. Now, that kind of leads to another question. The aspect of activated carbon itself allowing, in a sense, the diffusion of these products across a biomat that is formed on the carbon helps to ensure that you get less overall methane production at any point in time. So yes and no. If you break something down into methane, it's going to contribute. Most of it won't contribute because it'll be digested before it ever gets to the atmosphere. And the other aspect of it is, where else is it going to go? Hydrocarbons ultimately become methane one way or another. So they're hydrocarbons. So you're not making new methane. You are making new methane, but you're not making new sources of methane. The source is already there.
Right. Okay. Yeah, very interesting question. Here's another one for you. Why do we need to add an additional carbon source? Doesn't that just distract from hydrocarbon degradation?
Yeah, yeah. The additional carbon source, it was all the rage at Battelle this year, guys, at the Battelle Biological or Battelle Bioremediation Conference. All of a sudden, it's like everybody just discovered in the past year that, gee, maybe I should add an extra carbon source. It's funny how things come and go in popularity, right? It's so adding carbon sources is popular again. One of the reasons, what I pointed out earlier, is the labile carbon allows the microbes actually to take up more nitrogen, and that allows you to add more nitrogen. So one of the problems with just plain adding nitrogen is, there is a whole feedback loop within microbes for sensing nitrogen and what they want to do with nitrogen, and you can shut them down straight away if you give them too much nitrogen. The other aspect of nitrogen is that if you get the nitrogen levels right where the microbes like them, they'll use it up really quick. So one of the really interesting questions in bioremediation is long-term sources of nitrogen. So if any of you can develop a really long-term, see, in agriculture, it doesn't have to last very long, right? You want to put it out in the field, and basically you want it in those plants within a month. But in the subsurface, you want nitrogen that lasts for, 6 months, a year, 2 years? Yeah, you'd really like it to last for years if you could. One of the easiest ways to do that is just to build up a huge microbial population by giving them sugars and giving them the right amount of nitrogen and the right amount of vitamins. You can't forget the vitamins, they're unbelievably important. And what happens is, as they make amino acids and proteins in other parts of their body, they become the long-term storage source for nitrogen, because as they die, they release that, other microbes take that up. And so that's a means of obtaining a long-term source of nitrogen. So that's a big part of the sugar. The other big part of the sugar is that it has been observed, and not well explained, but observed, that you just plain get breakdown of hydrocarbons if you have more microbes, even if those microbes are not typically viewed as hydrocarbon degrading microbes. And there are a lot of hand waving explanations for that, but there are no really good explanations. So I'm going to give you one of those hand waving explanations, and if it's not a good one, well, you've been warned, okay? And that is that if you think of a microbe being in an aqueous solution, and with this contaminant, this hydrocarbon, that it may not like nor care about, they have a hard time keeping all of it out of their body. So we're not so different, are we? There are contaminants in our environment that we don't want, but we still have to breathe. So we breathe some of them in. We still have to drink water, and we get some. And the microbe situation is not so different. Now, when we breathe those things in, we address those contaminants in our liver by a whole series of various enzymes, the P450 reductase, glutathione, et cetera, quinone oxidoreductase, et cetera. So these are also, if you look in the little rollout that comes with the pharmaceutical you get from your doctor, you'll read about all of these various enzymes. Well, these enzymes didn't just pop up with humans. These enzymes exist in microbes. So glutathione, for example, exists in microbes. And so you will get these contaminants that are in a microbe that has no seeming direct pathway to degrade a hydrocarbon. And what will happen is a piece of that hydrocarbon will get conjugated to glutathione and spit out. This goes to the earlier question versus whether it's broken down or it just kind of, is inaccessible now. And that would be the second part of that question. It could be that some of the stuff just becomes inaccessible or no longer, you can't see it analytically anymore because it's conjugated to something like this. Now that causes me to say, okay, so that's what's happening. So you have this indirect pathway of dealing with toxic substances that are consistent with almost all microbes, that are even consistent with us, but they are not common pathways that people look at. And you'll get a lot more of those common pathways, or you'll get a lot more of those pathways as you increase the number of microbes.
Okay. There is a quick follow-up on the polar compound question. So in states like California, he says, in states like California, our regulators are concerned about processes that produce polar compounds, and we need to prove they are not harmful to the environment or human health. So maybe that gives a little more context.
No, it gives me a homework assignment, Ben. I have to look it up and figure out what California is talking about. But, I suppose if you could produce enough fatty acids that somebody might ask you, well, gee, are these things bad? But, in hydrocarbons, you have so many different substances. It may be some substance. Yeah, that's very interesting. I'll look it up and see what California.
Okay, well, then, Ben, you can watch for a response from Ed privately a little bit later then.
Yeah.
Okay, I've got one more question to throw at you. We've got about a minute before the top of the hour here. Is beta oxidation the only anaerobic degradation pathway?
No. Beta oxidation is the one that most people know well. It's the one that you'll often see put together in diagrams. But there are 6 other pathways. One of them would be the Wood-Jungdahl pathway example. And so you kind of have to keep that in mind, because sometimes, people will tell you that a particular metabolism can't occur because they're only looking at beta oxidation and they're not looking at one of those other 5 pathways. Sorry,
Okay.
I'm not going to go into it any more than that.
Yeah, no, that's good. Thank you very much. And really, thank you all again for taking the time to join us today and for all the really great questions. And I just want to encourage you to reach out to Ed Winter. He can flip down to that screen again, but you can reach him through the RPI website or email. And feel free to also reach out directly to me or others at AST. You can go to our website and there's project submission form. We do no cost evaluations of projects and can help identify what products and technologies might be best for your site. So feel free to reach out to us for that. And as I mentioned, you will get a recording of today's webinar in an email coming up here sometime pretty soon. And we encourage you to share that with colleagues, clients, however you'd like. And so I hope you all have a great rest of your day and week. And we certainly look forward to working with you at some point in the future. And thank you again for attending.
Okay, I appreciate you guys.